Hey, it's Joel Duff. Welcome back to my channel. Well, we've talked about, tuskless elephants. We've talked about mammoth meatballs, all in the past week. And I thought, why don't we just keep this Pachyderm party going? I saw a paper that just came out that reveals 23 new mammoth genomes. I don't mean really large genomes, although they're really large genomes. I mean 23 genomes of mammoths. Right, an extinct organism. We have 23 separate mammoths that have been sequenced now. Why do we need so many sequences? You might wonder that. It's like, once you have one mammoth sequence, don't you know something about what mammoths are? Why do we need to sequence more? That's one question we'll address as we look through this paper. I thought I'll just pull up this paper, we'll run down and look at some of the interesting stuff that came out of that. And along the way, as we look at why they sequence so many and why they're interested in looking at the population genetics of mammoths, we'll also be able to address some glaring issues with the young Earth creation model of post-flood speciation, and which is essentially population genetics over the last couple thousand years. And in particular, we'll see that Jeanson fails once again in one of his predictions. All right, so we've got 23 mammoth genomes looking at a brand new paper that just came out. Got that coming right up. All right, the task set before us is to take a look at this paper from Current Biology, Genomics of Adaptive Evolution in the Woolly Mammoth. So right there from the title, you'll see our genomics, we're going to look at the genomes of multiple mammoths. And by looking at multiple mammoths, we'll be able to say something potentially about their adaptive evolution, how mammoths have changed over time, specifically changed their morphology and other physiological features compared to other elephants. I mean, because if they didn't change them, they would just be like other elephants, right? But mammoths were different. So what makes them different? We're going to look into the genomes and ask ourselves, if we look at a bunch of Asian elephants and African elephant genomes, of which there are many genomes also, and then now compare it to these 23 mammoth genomes, what genes can we pull out from that we can see have changed over time and may contribute to the actual adaptive evolution of mammoths to their particular environment and time? So a typical article in Current Biology and a lot of research journals these days is to present what's called a graphical abstract. I really like these because it's sort of like, can you sum up your entire paper in one figure that gives you like the most pertinent information and hits sort of like the major bullet points? And then we'll take a look. So let's look at that. We'll also look at the abstract. And then I'm just going to scroll down through this paper and we're just going to pull out some interesting tidbits. We're even going to go into the supplemental material, which I like to do. There's often really interesting tidbits in the supplements. The supplemental information being like, these figures didn't really, they would take up too much space in the regular article. Like, we just want to have a concise article that says the main points. But there was also some other interesting things we found along the way. So we'll throw that in the supplemental material and give that to you as well. That's very commonplace now with many publications. Okay, so graphical abstract. So what we have presented to us here is we see this image of the history of elephants or pachyderms, right? And so we have woolly mammoths, Asian elephants, and African savanna elephants. We're going to ignore the African forest elephant in this. And it's been known for quite a while that the Asian elephant and the woolly mammoth are the most closely related, right? They're sisters to one another. And then more distantly related is the African savanna elephant. And they find that as well. So they compare a bunch. I'm going to show you the phylogenetic tree later, the more detailed one. This is the graphical simple abstract. And I'm going to show you that of all the sequences of woolly mammoths are all very similar to one another, just like the Asian elephant sequences are relatively similar to one another. And the African savanna elephants are more different than those two, which is why we would say that the evidence supports African elephants as splitting off from the common ancestor prior to the origin of the Asian elephants and the woolly mammoths. Then the other thing you see in the graphical abstract is you see these two boxes, and the two boxes include a bunch of names. All right, they're actually names of genes. And one box is highlighted, says genes highly evolved in woolly mammoths. And the other box is genes evolved in less than 700,000 years, over the last 700,000 years, basically. And so what they're indicating is from their analysis of 23 woolly mammoth genomes, which we'll look at and we'll see that those woolly mammoths span a fairly large and significant amount of time. The oldest woolly mammoth they have is 700,000 years old by conventional dating. And that elephant and all the other woolly mammoths all have significant changes in them to those particular genes that are listed. So you see some of those, for example, one of them is BRCA1, which is actually called the breast cancer 1 gene, same gene that's in human beings and other mammals. It's actually a gene for that protects one from developing cancer, and so errors in that particular gene can result in higher rates of cancer. Elephants are well known for their long life, large body size, and low amounts of cancer. So it's interesting that that particular gene is one of the genes that is highly, I'll say, significantly changed in some elephants. Anyway, the point is that all those ones in red there are all genes that they've identified by comparing the thousands of genes in the genome. And out of those many thousands of genes, like 20,000 genes, 20 to 25,000 genes, similar number of genes that you and I have. If you look at those 25,000 genes and you ask yourself, if I compare those genes in woolly mammoths, Asian elephants, and African elephants, are there significant differences in those genes? Are there significant variants that we can predict those variants cause different traits, right? There's significantly different versions of those genes that probably result in actual physiological, morphological, behavioral traits that are different between the elephants. And therefore, those are the genes that are responsible for making a woolly mammoth versus being an Asian elephant. And that's one of the things that they were interested in, is if you get enough genomes, get enough data to be able to compare a whole bunch of genes, you can identify the genes that are probably more involved in actually making the differences between organisms. So what they're indicating here is that list of genes are all ones that are all highly evolved in all woolly mammoths, and therefore they're saying very early on, after the split from Asian elephants, there was rapid changes in those particular genes. Rapid is a relative term, rapid over several 100,000 years, such that selection sculpted, that organism and sculpted those particular genes, changes in those genes to adapt elephants to a colder environment. And so as we'll see later, things like woolly hair, right? Thicker layers of fat, and there's other things that help them adapt to their northern climate versus the more southern climate of the Asian elephant. So they're indicating that these genes seem to all have already been in place. Like the oldest woolly mammoth that they could sequence already has changes in all those genes. And so presumably that's the reason they look like a woolly mammoth at that point. But then they look at more recent woolly mammoths than that older one, and they find that there's a whole nother set of genes that shows significant variations, all right? And we'll talk about what those variations are, what makes a significant variation versus a variation that is probably relatively meaningless or has no fitness advantage. They're saying these particular genes listed here are ones that they've identified as being significantly different than Asian elephants and African elephants. And so they have particular mutations, particular alleles, versions of genes that are probably helpful to woolly mammoths as they continue to change over time after that 700,000-year-old woolly mammoth. So they have a sequence from long ago woolly mammoth, all right, a member of a population of woolly mammoths from 700,000 years ago. Then they're looking at mammoths that are only 20,000 years old, 10,000 years old, 5,000 years old. And they're asking, how similar are those woolly elephants to the 700,000-year-old one? And they find is that, there's a whole bunch of other genes here that have changed since that 700,000-year-old woolly mammoth, showing continuing adaptive evolution occurring in the woolly mammoth lineage. Right, so that's the sum of the graphical abstract. Now we're going to flesh out some of the details as we go through the paper. I do want to give credit to the authors here. This has the ... here is there's multiple authors of this paper. And so let's look at the in brief. So we're not into the abstract yet. The in brief would be like the the abstract for the abstract. Again, in words, what is in the graphical abstract? So here we analyze unique non-synonymous mutations in 23 woolly mammoth genomes. Non-synonymous mutations are mutations that occur in genes that code for proteins. And non-synonymous means they make a change in the original DNA that eventually codes for a different amino acid. Different amino acids, which are what make up proteins, cause different shapes or conformations or charges in the resultant protein, and therefore can change the function of that particular protein. Not all non-synonymous mutations make changes in protein function, but many do. They're going to analyze which ones do, which ones don't, in order to figure out which genes are actually showing significant changes, like actual changes that matter versus changes that are just essentially neutral changes. So they look for non-synonymous mutations. We're going to call those are the important mutations that occur in genomes. At 23 mammoth genomes, so they have quite a number of entire genomes of mammoths, all right, representing mammoths from different locations and different times, including a 700,000-year-old specimen. So yes, we're talking about them extracting DNA from a specimen, preserved specimen of a woolly mammoth that is considered to be has been dated to be 700,000 years old. So this isn't the oldest genomic sequence of an organism. There's a horse that's at least that old. There's a couple other animals that have been sequenced that are close to around 1 million years old. But nonetheless, 700,000 years ago, yeah, that's nothing to sneeze at, right? That's a long time for DNA to be preserved. Now, that 700,000, all right, we're going to take a lot of tangents here, but that 700,000 year old DNA is clearly different than the 5,000-year-old woolly mammoth DNA from, say, Wrangel Island above Siberia. And by that, what I mean by that is more recent samples in general have much more intact DNA. The DNA is in longer strands. It is less degraded. It has less chemical damage to it. The 700,000-year-old sample has the most chemical damage, is in very short fragments. And yet they were still able to sequence enough billions and billions and billions of short fragments and able to compile those fragments together to reconstruct the chromosomes of that particular woolly mammoth. All right, so continuing on in brief. They find that woolly mammoths had highly evolved genes associated with hair, skin development, fat storage, metabolism, immune system function, body size, right? Woolly mammoths are huge. And some of those particular changes have only occurred in the last 700,000 years. So woolly mammoths are considered to be several million years old since a common ancestor with Asian elephants. But within the last 700,000 years, they've been challenged with various ice ages and so forth. So extreme conditions that have probably pushed them to continue to adapt their particular genes or continue to have mutations in genes and use those to adapt to those particular conditions. All right, so yeah, we're not even off the first page yet. I promise we're not going to be reading this whole paper. I'm hitting most of the really good stuff here, and then I'm just going to pull out a few specific things as we go through the paper. So again, we haven't even got to the very main abstract of the paper, but what are the highlights? Genomes of 23 mammoths and 28 extant elephants. So they also have 28 elephant genomes. Now that would be Asian elephants and African elephants. Not all the ones that they didn't produce all of these. Some of them were already in the data right out there. They've already been sequenced, but they added a few more to that database. So we're looking at more than 50 different Pachyderm sequences at this point. Interesting side tidbit. The way I found this paper is I follow somebody on Twitter that happens to be a postdoc that had sequenced 3 mammoth genomes and published a paper a few years back. And she's actually on this paper as well as a co-author. And what she noted in her tweet about this, when she mentioned this, she said, "Isn't it amazing that, a couple years ago, I got, basically a PhD at the time sequencing 3 mammoth genomes and be able to say something about mammoths. And that was a Herculean effort, right, to get your 3 mammoth genomes. And then along comes my successor right, who does the next PhD with that same advisor. And they go and do another 18 genomes, right, in less time, than it took to do those 3 genomes. That just illustrates the pace at which, or the advancement of DNA sequencing technology, such that, it gets ridiculous if you talk about what I did. For part of my PhD, I did sequence some genes. I sequenced RBCL, which is a 1,400 base pair long gene, and I sequenced five sequences from 5 different plants. One from each plant. So 5 total sequences, 1,500 base pairs each. And I spent, a solid year and a half working on that in order to get those sequences. I was super proud of that. And that was like a real highlight of my dissertation, was having a comparison of those 5 sequences, right? And now you can get an undergraduate in your lab and teach them how to generate several hundred billion base pairs of sequence. No comparison. Gene ontology, looking at how genes are used, suggests enrichment of mammoth genomic adaptations to cold environment. So as they look at all the different genes, and we know what most genes generally are involved with, because mammoths and elephants have almost all the same set of genes that human beings have. And so we have a pretty good idea of what genes are responsible for now. And so you can look at all these different genes, and you can ask where the changes are in the genes relative to Asian elephants, and say, okay, well, those Asian elephants and African elephants have similar genes for these things, but now the same gene that's found in the mammoth is quite divergent and has changed a lot. And so you ask, what genes have changed? Well, it turns out genes that you look at and you go like, well, what do they do? Oh, they're involved in skin and hair and fat production and so forth, right? And you start adding it up. You start thinking, oh, look, it kind of makes sense. Those genes are ones that have changed in order for those organisms to be able to adapt to their particular environment. Actually, I'm saying that incorrectly. The genes didn't change for them to adapt. The genes had variations via mutations, and the elephants living in that environment then were selected for the ones that had better versions of those adaptations, all right, or alleles that adapted them to that particular environment. All right, so they have highly evolved genes, including ones related to hair, skin, fat metabolism, immunity, and so forth. Several key phenotypes appear to evolve via heterozygous polygenetic selection. All right, now it's an important part of this paper. I'll make a stab at trying to explain it, but I'm not promising it's going to be clear. I'll take my first shot at it here. Polygenic selection. The word poly means many. Genic, genes. Selection refers to natural selection. So polygenetic selection is saying, look, most traits in elephants or you and I are actually made and constructed by many, many, many genes working together. And so if a trait is to change, if there's environmental pressure to adapt to, say, cold, right? You're living in a colder and colder and colder environment. Traits that help one adapt to the cold are going to be polygenetic in the sense that multiple genes are involved. And so you're actually requiring that over time, you're going to have changes in multiple genes that have an additive effect such that they add up to providing some kind of better fitness in that particular environment. So polygenetic selection is really a slow process, right? And that's going to be really important for some of the points I'm going to make later on. This isn't just like, there's this one gene and there was a mutation and all of a sudden, boom, all of a sudden they had long hair or something like that, right? This is hair is the growth of hair, the amount of hair, the thickness of hair, and all those things is determined by many, many genes. You're not just going to have single mutations or single variants that exist that immediately get selected for and suddenly change the hair. It's going to be a long, slow, drawn-out process of population genetics where you're going to have variations in the populations and you're going to have mutations caused by mutations. And you're selecting for a trait that is better cold resistance. And you're slowly going to be shape-shifting many, 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 many different genes over time. And heterozygous, hetero means different, and chronos refers to chronology. And so it's saying that At different points in time, you had different selection on different traits, and those traits are polygenetic. Right? And that's what they observed, and that was actually seen in the graphical abstract. All right? There's a whole bunch of genes that are selected for early in the history of woolly mammoths and have already been, we're going to call them fixed. Those traits are fixed into that population. And that's what makes woolly mammoths sort of have their unique characteristics. But then after that, at a separate time, there's been other selective forces in the environment that have also selected for a whole batch of different genes, for variations, for new variations in a lot of different genes that are continuing to adapt, all right, the woolly mammoth even better to its particular environment. And you can see how this kind of description of a paper is really helpful, rather than just like, okay, here's straight up a really complicated, jargony abstract like you might see in many papers in the past, and then of course the even more jargony paper itself. So let's get down to the paper. All right, so now we're at the introduction to the publication, Genomics of Adaptive Evolution in the Woolly Mammoth, and there's your long list of authors and where they come from. You'd imagine 23 genomes plus a bunch of other things they do in a variety of different kinds of analyses. They're leaning on the expertise of a lot of people who have contributed to this. And so it is a large number of co-authors. Summary: ancient genomes provide a tool to investigate the genetic basis of adaptations in extinct organisms. And that's one of the things that's exciting about this paper is we are looking not just at organisms that are alive today, but we're able to look at the history of organisms because we're able to go back in time and look at genomes. And that allows us to test some really interesting ideas that we have, or I'll say test a bunch of theories we have about how population genetics works, right? How about natural selection works, about how genetic drift works, right? All the mechanisms of how organisms change their genomes over time. And so we can look at those and we can do short-term projects, right? And we can manipulate things and do natural selection and artificial selection environments and watch genomes change from a couple generations. But if you're able to go back thousands of generations and then compare that to descendants 1,000 years later, you're able to look at more of the full scope of the trajectory of genetic change in a particular species and be able to test our ideas in different ways. So, however, the identification of species-specific fixed genetic variations requires the analysis of genomes from multiple individuals. What are they talking about here? The identification of species-specific fixed genetic variants. All right, fixed genetic variants are individual alleles, variations of genes, right? You've got, you have a gene for, my favorite one is middle digit hair, or not having middle digit hair, right? You have one gene, that gene has different variants, and depending on which combination of variants you have, you'll have that particular trait when it's expressed. All right, so there are different variants. What a fixed genetic variant is would be saying that if every human being had middle digit hair, because that was the only allele, that was the only variation of that gene. In other words, we're all homozygous, right? We all have two copies of that. We got a copy from both of our parents and it's the same. Then it would be a fixed variant in the population. And it's a variant because we're referencing all the pachyderms or all the elephants. So compared to Asian elephants and African elephants, they have a different allele. They have the same gene, but they have a different variant that does something slightly differently, right? The program is slightly different. The protein it makes is a little bit different. And the mammoth has fixed genetic variants in it. And so these are species-specific fixed genetic variants, meaning only mammoths would have these particular variants. Now, let's say you only have one sequence. Right? You only have one genome. You worked really hard. You got one genome, and you can compare it to other organisms. And you see that one individual has middle digit hair. All right? And another species doesn't have middle digit hair or has some variation in middle digit hair. How do you know that that particular characteristic is fixed in that particular species? All you know is that that individual has that characteristic. But what if you were able to look at 20 other individuals from that particular species? How do you know that they don't have variation? Is it fixed in the species? Do all members of the species have this particular variant? and therefore it's characteristic of the species? Or is it simply characteristic of some individuals in the species? The only way to know that is you got to have multiple samples. So this is one of the reasons why you try to sequence as many copies or many individuals as possible, and you need to get a nice breadth of individuals. You can't just be like, I'm going to go to one location, I'm going to find 23 mammoths that all died at this one location. Well, they may all be part of the same family, right? Or at least they're all part of the same population. If they're part of the same population, they might have a fixed variant. They might all have a particular variant for blonde hair, say, or red hair, or extra thick skin, or whatever, right? Would that mean that all mammoths have that particular characteristic? I wouldn't necessarily be able to extrapolate that way because I don't have a sense for what the genetic variation is within that particular species. That's what we're trying to accomplish here. What is the extent of genetic variation in a species like this? So what you do is you go out and you sequence as diverse a population of individuals as you can to capture the total diversity of the species. Now, they haven't accomplished that with 23 sequences, but what they have done is they've not only found samples from different locations, all right, that are hundreds of miles apart, and therefore probably parts of different populations, but also the individuals are from different time periods, right? So you're looking at, so if you have something that's 700,000 years old, 50,000 years old, 40,000 years old, 30,000 years old, 15,000 years old, and a couple individuals that are 5,000 years old from different locations, and they all have exactly the same variant, and it's different from Asian elephants and African elephants, well, that's pretty good evidence that that's probably a fixed variant and a meaningful, significant variant to that population that contributes to the appearance or the nature of woolly mammoths compared to other types of elephants. All right, so yeah, we just got through sentence number 2. All right, third sentence. Moreover, the long-term scale of adaptive evolution coupled with the short-term nature of traditional time series data has made it difficult to assess when different adaptations evolved. Exactly. I mean, if you're only looking at a few generations, and you might look at a bunch of individuals in a population or in a species and see that there's differences, and then you're asking, where did those differences arise? You can kind of maybe extrapolate where in history those variances came about, but it's hard to know when those adaptations happened. Whereas in this case, since you're actually looking at historical samples, right? You're looking at ancient samples. You can assess all the samples from different ages, and you can see how maybe there's particular mutations, right? Particular variants that entered into the population at some point. And then after a while, all the individuals have that particular variant later on in time. And so you can point to some window of time and say, like, that's when that animal found that adaptation, right? That's when that adaptation was occurring, for thicker blubber or whatever, all right? Longer hair. And so they get some kind of snapshot of the timing of changes in organisms. All right, so here we analyzed 23 woolly mammoth genomes, including one of the oldest known specimens at 700,000 years old, to identify fixed derived non-synonymous mutations unique to the species and to obtain estimates of when these mutations evolved. Can we figure out where those mutations entered into the population? This is also going to be important when I get to sort of my application of this to sort of the world of creationism and their model of population genetics. The idea that if you're looking at different ages, different time spans for woolly mammoths, and if you look at the older ones and they don't have a particular variant, and then it's only recently that a particular variant occurs, that's one way of saying that that's a mutation that entered into the population. It's not an original variation that existed in the ancestor of all of them, because we don't see it in any individuals in history. We only see it in more recent or long ancient history. We only see it in recent history. Okay, so what did they find? We find that at the time of the origin the woolly mammoth had already acquired a broad spectrum of positively selected genes. All right, at that 700,000 years old, when clearly mammoths are already there in their, basically the same overall appearance that they have later on. They already have a bunch of positively selected gene variants that have been selected for different from Asian elephants and African elephants to give them their unique traits, including ones associated with hair, skin development, fat storage, metabolism, and so forth. Our results also suggest that these phenotype refers to appearance of characteristic, as opposed to the genotype, which is the genes that underlie that characteristic, continued to evolve in the last 700,000 years, but through positive selection on different sets of genes. So interestingly, they find that the changes in that original set of genes don't continue to occur. So those genes stay relatively static from 700,000 years ago all the way to present. The genes which show big differences between other elephants have, you could say, they've reached their maximum potential for change within that particular environment. And now they're no longer being selected for any additional changes. So what's happened is there's other genes in the genome now that are being selected for adding other traits to the mammoth. So the mammoth is acquiring new characteristics via other genetic pathways, not the original ones that they used to do the first part of their adaptive, become the species they are. Finally, we also identify additional genes that underwent comparatively recent positive selection, including multiple genes related to skeletal morphology and body size. In other words, there is, by the way, morphologically from the fossil record, we also know that there are changes over time in the mammoth body, and that's also shown here in the genetic data now that they have these genomes, as well as one gene that may have contributed to the small ear size in late Quaternary woolly mammoths. Late Quaternary would be like very recent woolly mammoths had some of what appear to be the smallest ear sizes, and maybe they found a gene that's related to that. Okay, so that's the paper. We're not going to read all this background. We're just going to go down to the figures. So I wanted to show you this one. This is a phylogeny in circle format or radial format. And what they're showing here is the mastodon. There is a mastodon sequence as well, and mastodons are extinct. Mastodons, by the way, are very different genetically, clearly highly genetically distinct, all right? Genetically divergent, distantly related to the African elephant, Asian elephant, and woolly mammoth. So we consider them as part of the family of pachyderms, Elephantidae, but they are much more ancient themselves in terms of where their common ancestor would be found. All right, so then all these purple ones up here, these are all individual African elephant sequences that have been generated. All right, and roughly speaking, so they're showing the relationships of those, how similar they are to one another. And then all of them are related by a common ancestor, showing that African elephants are all the sequences are generated, can be shown to have a common ancestor, although there's quite a bit of difference between some of these sequences. And then we've got the Asian elephants over here. And again, all the Asian elephant genomes that have been sequenced are similar to one another, but not the same. But nonetheless, they all form a group, and we call that a monophyletic group because they share a single common ancestor. So if you go back in time, there's a single population, or there's not a single individual, but there's a single population of elephants that share a very similar genome. And then over time, those elephants have diverged into more divergent genomes over time, but they still remain similar enough to each other that we call them Asian elephants, and we haven't separated them into new species. Now we get to the woolly mammoths. And so now here's our 23 genomes of woolly mammoths. How about that? We've got more genomes of extinct organism than we have of living animals today. But these questions are really much more interesting because of the time aspect, able to go back in time. And this one right up here that's oh boy, I should never try to pronounce things. Chukochya, that is the one that's 700,000 years old. And it is the most, what we call basal, or the most similar sequence to the Asian elephants. And then all the other mammoths, which are younger, are basically have more differences, all right, to them that make them more different than Asian elephants, right? And you can think of that as through time, you're going back 700,000 years when you look at that particular sample. The other samples you're looking at more recently in time, and therefore there's been more time to accumulate more mutations, and therefore they're going to look more different, right, over time than the other ones. So there's all kinds of talk about they go through each of the different genes. They talk about how each of these genes has been affected. And then this is just a summary of like the genes we identified in terms of our analysis of all the different genes and their functions, right? And we asked, are there significant differences between them? We find that the genes are involved in these types of things, right? Protein, sugar synthesis, cell structure and function, and so forth, right? And I just wanted to show this picture too, so you can see that an Asian elephant and a woolly mammoth, although they have similar body styles, all right, you can see the sloping back on a mammoth, right? The tusks are different, the ears are different, right? The head's different. They have this large amount of fat storage. And that's also going to protect them. This helps for overwintering, but also helps for protection against the cold and so forth. So there are different types of organisms, all right? They're not, they have similarities, but they're distinct in many, many different ways, and so clearly identifiable as a separate species. Okay, keep on going, keep on going, keep on going. All right, so now here we get to number of FDNS, and FDNS refers to fixed non-synonymous variations, all right, or variants. Fixed non-synonymous variants. So across here we have 0 up to, I think this is 19. And so that's the number of individual fixed variants in a particular gene, right? So as we look across the genome and we look at sections of the genome and say, like, here's where a gene is coded, and a gene might be hundreds to tens of thousands of base pairs long. And you look at the variants that are there, the differences between this woolly mammoths and Asian elephants and African elephants. And you asked which ones are fixed. Now, fixed means that all of the different woolly mammoths they look at all have this particular variant in common. So there isn't a mixture of like, oh, there's a few Asian elephant variants mixed in that population. No, it's all the same variant. And there's a lot of variants in that particular gene. So, for example, this way up here, AHNAK2, I didn't look up to see what that is, what that particular gene is. But it has 14 or 15 individual variations, which would be A's, T's, C's, and G's that are different than all other elephants. So they're fixed in all the members over all time that they looked at. And those are all non-synonymous changes. Non-synonymous, again, I want to remind you, means that there's a change in the A, T, C's, and G's, but it's at a position that when it's read into a protein, will read a different amino acid. And that a different amino acid then has the potential to change the shape and structure and function of that particular protein. So how do they know, though, whether it changed the function of the protein or not? All right, there's lots of sophisticated ways to do that. Lots of ways to do that, but here's the simple way they've done it. They've scored it. They've given it a score, and I can't go over like how that happens because I'm not exactly clear, but I know what the general gist of this is. The general gist of this is they give a score to the different variants as to how significant they think that particular variant is based on lots of studies of that particular gene and other variants maybe in other organisms and also knowing something about the particular site. Like if you're changing the active site of an enzyme, you know it's going to make a real change in how the enzyme functions. Right? So you're scoring all those 14 variants and you're saying if you get a really high score for this, that means a lot of those variants are making that protein be very likely to be doing something very different in the mammoth versus what it's doing in other organisms. So they have the same gene, but they're doing different. They're having a different outcome, right? A different phenotype, right? A different appearance as a result. And you'll notice there are some genes, like BRCA2, which is an oncogene, right? It's a gene that helps prevent cancer, has experienced a large number, 19 non-synonymous changes. But they, in their analysis, have determined that it's unlikely that many of those or all of those variants have any significance to the function of that particular protein. And so all these ones in red are all fixed variants that they know the names of particular genes, and they know that those particular genes are showing real dramatic differences in their function. And then they're going back and saying, well, what do those genes do? And what makes more sense here is that these aren't just a random set of genes. When they start to look at those genes, we've already mentioned, those genes are involved in hair, skin, fat production, right? One of them is the ear size developmental gene and so forth. So they make sense with respect to like, oh yeah, I could see how this gene could play an important role in changing and adapting an organism to a cold environment. Okay, now these are fixed variants. It doesn't mean there aren't other variations, right? There's going to be synonymous changes, but the synonymous changes don't change the protein function, and so we don't really care about those. The fact you get 10 or 15 non-synonymous changes in a particular gene, I got to tell you, that's really significant, right? And just to illustrate that point, there are only a few genes in human beings compared to, say, chimpanzees that show a significant number. I mean, numbers are usually smaller than this in terms of the number of non-synonymous changes that are in particular genes, showing that those two genes have different functions. All right, so if we were to map other species, we'd find there's always going to be differences, and there's going to be significant differences in some genes. But this is a lot of differences, all right, between these two, showing that there is a high amount of selection pressure in that particular environment living up north that has rapidly selected for mutations at non-synonymous sites, selecting them and allowing them to move to the next generation, and eventually all elephants have those particular mutations and they become fixed in the population. All right, so that's fixed. This is the list of genes, and over here is the predicted phenotype affected. And so fixed derived synonymous, fixed derived non-synonymous. It would see like fixed derived synonymous would be like synonymous, they don't actually do anything to the organism in terms of its function. They could still become fixed. They just won't become fixed by natural selection. They become fixed by the forces of genetic drift and so forth. And so having a bunch of fixed derived synonymous, and derived, I need to point out the word derived also means because they're not found in the other elephants, they're derived, meaning they're new mutations. New mutations that have entered into the population that gave rise to woolly mammoths and then became common enough that they became fixed in that population. But what I want you to notice is if you go down this column, if you go down the column of fixed derived synonymous, the numbers are smaller in almost all cases than the fixed derived non-synonymous, right, for these particular genes. That indicates that there's natural selection going on, and the natural selection is fairly strong, selecting for these important variations that change function. If I were to show you, if I were to pick out a random list of genes from elephants and from woolly mammoths, and we were to look at the number of synonymous versus non-synonymous changes, typically what you would see in most cases, like comparing me and you, or even me and you to chimpanzees or something like that, you would find that you'd have more synonymous changes than you would have non-synonymous changes, because it's actually easier to make a synonymous change. More mutations cause synonymous changes, and so you expect more synonymous changes in between genes as they're diverging, simply because it's easier to happen. So when you see more non-synonymous changes, that tells you there's a heavy hand of natural selection that is selecting for those, causing them to be more numerous in future generations. And that's how we also have a really good feel for, and feel for is not the right word, why we have a lot of confidence that these particular genes and their functions are playing a really important role. These are the roles that have adapted woolly mammoths to their particular environment. All right, past the references, of which there are many. And we're going to get to the supplemental material. All right. Oh, yeah, we're still in protocols. If you want to know how they did it, here's all the different methods and materials, which in this particular journal, they're actually putting after the references. So it's kind of like, you scroll way down there if you want to find out like what exactly we did. So now we get to supplemental information. And it's the same set of authors. They've also provided this additional information. There's only 2 figures, and we're going to look at the first one. All right, so here's the first figure. And the first part is this. They're just showing you graphically, right, or on a chart, the coverage for the genomes and the time. And I'm really interested in the time here. So these are different locations. The different colors are primary locations of where the samples come from. And then the time on the bottom is in millions of years. I'm sorry, not millions of years, thousands of years. So 5,000 years, 10,000 years, and over 50,000 years. So the one that's over 50,000 years is 700,000 years old. And these other dots represent individual samples that were sequenced, and you can see how old they are. So they have a nice distribution from 5,000 years ago to about 45,000 years ago, all radiocarbon dated. And then you've got what's called coverage. What does coverage refer to? That's actually referring to the number of how many times they sequenced the genome over. All right, so or how much total data did they get for that sample? 10 times coverage, right? 10x coverage is what that means. 10x coverage would be they've sequenced enough of the genome from multiple samples of cells that they have effectively sequenced the entire genome 10 times. And then they stack those 10 genomes on top of each other. And then they compare the sequences of those 10 genomes. And what they're doing is that helps to avoid errors, right? There's going to be mistakes made in the sequencing process. So that an A and a T and something like that once in a while is going to get chucked in the wrong spot. Or there's going to be damage to the DNA somewhere in one cell. But it isn't necessarily damage in another cell. So when you sequence it from one cell versus another one, you're going to have little nicks and bad spots. If you have 10 copies of the genome and they're all laid on top of each other, you can go through and say, well, you know what? 8 out of the 10 have this particular sequence, and these other 2 have missing base pairs or base pairs we couldn't read. So we can fill in the gap using the other 8 that we know about. So same thing would happen if you sequenced U, all right? We would sequence not just 3 billion base pairs, we'd sequence 100 billion base pairs. And then we'd overlap all those sequences with each other in order to get your correct sequence from the consensus of all those sequences. All right, so that's what that's about. We'll come back to the dates in a few moments. All right, what I'm really going to, right? Actually one of the reasons I even doing this video at all is to talk about this figure. Here we have heterozygous sites. Heterozygous sites. All right, so heterozygous, remember, refers to elephants are diploid just like you and I. They have 2 copies of their genome. They got one from their mom, one from their dad. So you have 2 copies of every gene. And so for each gene, we've been talking about variants, right? And we're talking about variations. And we've talked about fixed variants, saying that if you're fixed, it means you have the same variant from your mom and from your dad. But not all of your genes are completely fixed. There's lots of variation in a population. Obviously, not every single woolly mammoth is identical, just like you and I aren't identical. So we have many fixed variants between us, and they're fixed between all human beings, some of our variants, many of our sites in our genome. But of course, there are places where we're not fixed and where there's variation. And so potentially you have a different variant from your mom and your dad. So if that's the case, you're heterozygous for that particular variation in the population. So what this particular figure is showing is the total amount of heterozygosity that was identified among all the different elephants, all right, or within those elephants. All right, so if you look at the 700,000-year-old woolly mammoth, and you look over here at this 1.5 times 1,000 base pairs, so 1,500 individual heterozygous sites of the different variants that they were looking at in the genome. And look at the 45,000-year-old one, 1,500 variants, 40,000 year old, and so forth. You go across, this is generally pretty flat, meaning there's about the same amount of heterozygosity, the same amount of genetic variation existing 700,000 years ago all the way up to 10,000 years ago. Then there is this one red dot here, which is significantly lower. That's the Wrangel Island samples, all right, or sample. The Wrangel Island sample is only about 5,000 years old, or just a little less than 5,000 years old. And those are considered to be the last of the woolly mammoths. Those are the woolly mammoths that you may have heard of that got stuck on a little tiny island way up north of Siberia, and it was like the last hangout after all the other mammoths were wiped out. At least that's as far as we can tell. There was one hanger-on population, and on that island, they lived on that island for many thousands of years, isolated there. And in that isolation, they had to breed with each other because they didn't have a very large population, so they're highly inbred. Inbreeding results in homozygosity. It drives out heterozygosity. And so that would actually be a prediction you could make before you actually even sequence the Wrangel Island woolly mammoth, is if it's true that they were on that island for a long period of time in a relatively small population, they should be losing their heterozygosity, and they should be moving toward more homozygosity. And in fact, that's exactly what we see in the genomes, is we see that they lack heterozygosity like the others do. So that's kind of a special case of reduced population size, isolation, inbreeding, and they also, by the way, got reduced in size as well. So they had a lot of selection, a lot of selective pressure to change their morphology in order to adapt to living on a smaller island with fewer resources. And so they dramatically changed over time. All right, so now why am I saying I brought this up? What does this have to do with anything with respect to creationists? All right, I've already gone a long time, so I'm going to try to make this kind of a little shorter. Young Earth creationists have a model in which, rather than saying what I've been saying during this whole talk, which is that woolly mammoths have fixed variants, and then over time they fixed even more variants in other genes. But where did those variations come from? We would say that they come from new mutations, right? Every time elephants are copying themselves, they're creating new variants. And so they're creating new heterozygosity because they're creating new variants that aren't fixed in the population, so therefore individuals could have one variant or another variant. And so that increases the number of variants in a population. And then those variants, of course, could be selected upon if they have an advantage. And so where did all these new adaptations come from that made what woolly mammoths are as a species, adapted to cold climates? We'd say that many of those characteristics, right, are derived from a population of elephants that's dividing off from Asian elephants and moving somewhat farther north, experiencing colder climates, and they're having mutations. As they have mutations, that colder climate selects for mutations that gave benefits in that particular environment, and eventually all the elephants had those particular variants because they were beneficial and they became fixed in the population. So what I'm saying is, those variants didn't always exist. They don't exist necessarily in the common ancestor of elephants. If you go back to Asian elephants, and then you go back to African elephants, there's a common ancestor at some point, right? Let's go back up here. Right, if you go all the way back to the point where the common ancestor is that gave rise to Asian elephants, African elephants, and woolly mammoths, we'd say that all the variants that you see today in Asian elephants, woolly mammoths, and African elephants, all those variants didn't all exist in the ancestor. Some of them did. There would have been a population at that point in the ancestor that had variation. Some of that variation is going to be selected in different populations to become some parts of those new species. But along the way, in those new populations that are diverging from one another, there's going to be mutations. Look at those genes that are highly evolved in the woolly mammoth, and look at those ones that seem to have appeared, new variants that have appeared since 700,000 years ago. Those are variants that didn't exist 700,000 years ago in that woolly mammoth, likely didn't exist in that population, although we'd have to have more samples to prove that. But almost certainly didn't appear in the ancestor of Asian elephants and woolly mammoths. Then you go farther back to Asian elephants and even, African elephants, and many of those variants that existed that are shared between Asian elephants and woolly mammoths, they don't exist in that common ancestor. So what we're saying is there's new variants occurring all the time, and those variants are being used for adaptive evolution. How is that different than the young Earth creationist model? Okay, I'm going to show you a paper from Answers in Genesis from a few years back. Does biologos strive for dialogue? It doesn't sound like it would have anything to do with this, but what this is a, how shall I put this? This is Dr. Nathaniel Jeanson in 2016, and it's really a diatribe about me, complaint about my writings about him and some things I've said about young Earth creationist views of speciation. All right, so all of this stuff is about Dr. Duff in here, and so I wanted to scroll down to the relevant portion. Okay, I want to scroll down to this portion. He quotes a section from a blog post of mine from the year before. And so I'll read this portion, and then we'll look at his response to that. The origin of species from a common ancestor on board the Ark occurred by evolutionary processes. This is made clear by an exhibit at the Ark Encounter where we learn that species give rise to new species. Modified characteristics develop over time, and the fittest animals survive. Right? That's a quote from the Ark Encounter, a panel at the Ark Encounter. This is a reasonable description of Darwin's explanation for the origin of species by means of natural selection. Right? I'm saying they're describing natural selection as Darwin laid it out. Many visitors to the Ark Encounter might be surprised to discover exhibits which embrace Darwin's mechanism, natural selection, for producing new species, albeit with some strict limits. Along with other natural mechanisms of change, the Ark Encounter has proposed what I call post-flood rapid speciation model, or modern biological diversity, to solve the problem of limited dimensions of the manpower of the Noah's Ark. Right? They only have so much room to put all the animals on the Ark, so one thing you do is you have all the canines be one kind, and then you just have two canines on the Ark that became all the canines today. So many different species. How does that relate to the elephants? Young Earth creationists believe that there's only one kind of elephant, one created form of elephant. So there were two elephants on the Ark. Those two elephant-like organisms on the Ark stepped off the Ark, and then they had offspring, and those offspring then created populations, and those populations eventually diverged into woolly mammoths, Asian elephants, African forest elephants, mastodons, platypedalodons, Dinotherium, and the other 100 species of extinct elephants that we know about. Right? And all of that had to have happened in the last 4,350 years. In fact, in a much, much shorter time than that, because those 100 different species of extinct elephants are probably all extinct before the Ice Age. And for young Earth creationists, the Ice Age is just 400 years after the flood. So in a space of just 400 years, you have the development of maybe 100 different species of elephants. There's all kinds of problems that you could imagine with that. But let's just stick to the population genetics here and what we've been talking about here with heterozygosity. So in their model, heterozygosity is key because they don't want to admit that new mutations are really responsible for adaptation, for making new species and new characteristics in species. They want to say that God placed in the original kinds of organisms that he made all the genetic variation that was needed. So therefore, their model is that there was a tremendous amount of genetic variation, and that variation is being essentially sorted out into different distinct piles or distinct sets, which we now call species, right? Because you took these variants and kind of like selected for them in one environment, took another set of variants, selected them for another environment, and then you get two different sort of looking types of organisms that we call different species. But in fact, they're just sorted from an original variant pool. I got ahead of myself. We're going to explain that in here. In reality, creationists have embraced natural selection and its profound limits for decades. That's true, and I'm not denying that they haven't. They haven't done so in such a public way as the Ark Encounter does, which is why when I go to the Ark Encounter, I watch people be really surprised by what they're saying there. So they've not been as open about their embracing of natural selection. And if you watch any of my other videos, you know that not all young earth creationists embrace natural selection. Some are vehemently against natural selection, despite what Nathaniel Jeanson is saying here about how we've embraced it. It's been a textbook teaching point for Answers in Genesis and YEC scientists for years. The only people who will be surprised are those that have paid little attention to young earth creationism. I guess that is most people that follow young earth creationists as the lay people. Of course, he's actually referring to me, saying, basically accusing me of not paying attention, which is kind of ridiculous considering I've probably read more young earth creationist literature than Nathaniel Jeanson probably has. Furthermore, we're getting to the point here. Furthermore, Duff's blanket statement above reveals his unfamiliarity with the YEC technical literature. This origin of species from a common ancestor on board the Ark did not occur by evolutionary processes. Do we invoke natural selection? Sure. All right, that was a quote from me. Sure we do. The placards right next to the one that Duff photographed and included in his article explain our position clearly. So what is Duff missing? A gigantic difference between young earth creationism and evolution traces to the source of genetic variety we see today. As I published previously and discussed in great depth, he likes to talk about how he's done everything in great depth. Darwinists think that all genetic differences are due to mutation. Boy, I'm tempted to go into a diatribe about the misuse of terms here, but okay, let's move on. Darwinists think that all genetic differences are due to mutation, right? Genetic variations in populations, the result of mutations that occur as organisms unfaithfully copy their genome to the next generation. I have argued, now here's where Jeanson's going to take issue. I have argued that most differences between species are due to shifts from heterozygosity to homozygosity. A technical term, I guess he's referring to the word homozygosity. I mean, I suppose that's technical to somebody who hasn't gone to high school, but homozygosity is a pretty, like one of the most basic genetic terms. A technical term implying that God created kinds with built-in genetic diversity. I'm not sure exactly what the technical term is referring to here. Shifts from heterozygosity to homozygosity? Yeah, now I'm actually confused. Homozygosity? But how is that a technical term that implies that God created kinds with built-in genetic diversity? All right, but point is, he's saying that God created kinds with built-in genetic diversity, massive amounts of genetic diversity. In other words, high heterozygosity. Like if we had the original kinds, their heterozygosity levels would be like through the roof. And then what he predicts you would find then is over time, as you procreate and as populations separate from each other and natural selection selects for variations, it's obviously going to select out some variation and it's going to create more homozygosity, more fixed variation, fixed variance, the fixed variance we've been talking about. And so he's thinking that you're going to start out with a huge amount of heterozygosity and then you just winnow it down. And so you shift from heterozygosity to homozygosity. And so if you looked at the ancestor and then looked at the descendant species. So in our figure there, there's an ancestor to Asian elephants, modern Asian elephants, and mammoths, right? And if you went back to that original population before it split into those two separate populations, he would predict that there was more heterozygosity there, right? More genetic variation. And then that genetic variation that was in that pool of genetic variation got selected for, and some of the variants were selected for to make Asian elephants, and different variants were selected for to become mammoths. But as a result, you would have less total variation in each of the subpopulations. Maybe if you added the two populations together, you'd have the original variation, but you can't possibly have as much variation in the species that are the products of an original common ancestor. Now, this kind of sounds intuitive, I'm sure, to a novice, to somebody who is naive about biology and the data from biology. they might be tempted to think that this sounds intuitive, like, yes, of course that's going to happen. And in some ways it does, right? There are definitely species that are spinning off. There are populations that spin off of larger populations, and they only carry a small subset of the original variation. And the new species that is eventually generated from that will probably have less total variation than that original population does. Will it have more homozygosity? Not necessarily. But let me continue here. This singular difference in the source of genetic variety has massive consequences for how new species can form and for the limits of speciation. See, he's thinking that since you have an original pile of variation and then it gets sorted out, then the species that are generated don't have as much variation, so they can't create as many more species, right? Because at some point, you can't split the species over and over and over again. You run out of variation. If you run out of variation, there's nothing to select for, in which case organisms can't adapt anymore. They cannot adapt to their environment because they have no variation. And therefore, they will only ever stay as they are. And so he sees the world as essentially bifurcating populations to the point where eventually there will be no variation within species. And then no new species could be formed, right? So that is the limit on speciation. Unfortunately, he runs up into reality. All right, the reality is that's not the observation from the world, right? You can force yourself to see this happening somewhat, maybe if you look at artificial systems like domesticated dogs, right? Where we've bifurcated the genome of wolves into separate little piles, which are all homozygous. Like dog breeds are extremely inbred, have lost their genetic variation, and they do have extremely low levels of heterozygosity, right? But those are very specialized cases. When we go like the natural world, what we find is we find that many species, large numbers of species, have generally similar amounts of heterozygosity. So remember, I said that these elephants, they got off the ark, right? If they got off the ark and then became a population, and that population split into 10 populations, and each of those 10 had selection on them, and they lost a bunch of variation, and so they went from heterozygosity to homozygosity, and as Jeanson says here, the differences between species are due to shifts from heterozygosity to homozygosity. Then after you've split a few times, there should be a lot less heterozygosity. And then, but the thing is, you had to keep splitting those elephants, I guess, in order to get 100 different species of elephants. But what we're finding is, as we sequence 4 or 5 different species of elephants with multiple individuals, we're finding actually very similar amounts of heterozygosity in each one. And very appropriate amounts of heterozygosity that would continue to allow speciation to occur. And here's the key. In the case of woolly mammoths, because we have we have samples over time, we could ask how much heterozygosity was there 700,000 years ago versus recently. Because he would even argue that within a species, you're constantly losing variation because natural selection is removing variation, and even genetic drift does as well. And so there should be continual losses of genetic variation. This whole idea of kind of goes along with genetic entropy, where organisms are falling apart, but genetic entropy is actually mutations that are adding variation, which is like almost the opposite of loss of heterozygosity for Jeanson, but I'm not sure how he melds those two models together. It could be he's not a big fan of genetic entropy, but many people at Answers in Genesis are. All right, sorry, tangent there. All right, let's go all the way down here. Here we go. So remember, what I'm showing you here is that heterozygosity has remained relatively constant over 700,000 years. All right, there's not a loss of heterozygosity. Now, populations are constantly losing variance, right? As organisms reproduce and as natural selection sweeps out inefficient alleles. The thing is, there are always new mutations, right? Mutations are recreating variation all the time, right? Reestablishing variation. And so even when I mentioned that sometimes you have a spin-off population that gets isolated from a larger population and eventually might become a new species, and I said they'll have lower amounts of variation. And because they have to inbreed and because they're small populations, they could have lower amounts of variation. What you'll find is over time, they increase the amounts of variation. All right, cheetahs have like almost no variation because they've gone through a bottleneck, and they were reduced to very, very small numbers, forced to reproduce with one another. That did take heterozygosity and turned it into almost all homozygosity in those individuals. But over the years, they're adding new variants to that population, and they are becoming more heterozygous over time. Now, it's going to take thousands of years for them to re-establish the amount of heterozygosity or genetic variation in their population that they once had. But it is inevitably moving in that direction as long as that population continues to persist and grow in size. And so watch out for young Earth creationists that may point to like exceptions to the rule in order to try to prove a rule. All right. Their rule being genetic diversity is lost, right? That heterozygosity becomes homozygosity, that the original creation had massive amounts of genetic diversity and it's only being lost over time. And so you can find individual samples like the mammoths on Wrangel Island or the cheetahs, or, you can point to various things like that and show like in the past they had more variation, now they have less variation. But that's not the rule. That's not the general rule. That is the exception to all the other data in the literature. And here we have another example. Here we have an example of woolly mammoths, which have maintained genetic variation for long periods of time. And if you compare this to Asian elephants and African elephants, they also have a good amount of genetic variation still, despite the difficulties in their populations. So we do not see this trend of loss of variation. And we have other examples where you can go back and look at something that's close to the common ancestor and actually estimate the amount of variation in those. And the variation is not that much different than the descendant species. And again, as soon as new species are formed, new variation continues to be pumped into that population through mutation. So I don't know how he can deny, he's saying that all the important variations exist in the original creation. But it's quite evident that many new significant variants that add to the adaptability of organisms and therefore are beneficial mutations, they occur, and they're mutations and they're changes compared to the original state of the population. Oh, that reminds me, if you think about the tusklessness nature, at the end I kind of speculated about, if you have this gene and there's two different alleles and one of them is for tusklessness, either young Earth creationists have to say that's a mutation, except that it's actually a positive mutation in particular selective environments, in other words, where tusks are disadvantaged. So tusklessness is not necessarily a bad variant. So did God create that particular variant in the original creation? Or is it a new variant as a result of mutation? And I think Jeanson doesn't want to call that mutation, but I think he's going to have to recognize that some of those things are mutations. You might want to call it bad, but it's going to be inescapable that there are positive mutations that occur. And so changes have occurred since the original creation. All right, so this is what I call, this is a failed prediction of Jeanson's model. Now, Jeanson might complain. He might say, look, I don't believe that's 700,000 years old. I don't believe your timetable. But he does actually believe what does 50,000 years down to 5,000 years represent in his time frame? Remember, he believes that all these animals must have lived since the last 4,350 years. All right, so the oldest date, the 700,000-year-old one, must be something that is like something like that. That represents an elephant that must have gotten right off the ark. There's another problem. There's clearly a woolly mammoth. And that woolly mammoth is dated to be 700,000 years old. In his particular way of converting radiometric dates into biblical dates, 700,000-year-old date would be something that is in the first 100 years or so after the flood. So he's going to have to propose that animal got off the ark, an elephant got off the ark, and immediately there was a woolly mammoth that existed. I guess you could say the woolly mammoth is the one that was on the ark, and then all the other ones evolved from that one. Then the other issue here is he converts carbon-14 dates. He doesn't say that carbon-14 dates. This is a newer way that young Earth creationists and Answers in Genesis in particular approaches carbon-14 dating. They don't say it's totally bogus, right? There's obviously different amounts of carbon-14 in these samples, and they follow a pattern, right? In sediments and so forth, it's like less carbon-14 and more carbon-14. as you go up. So there's an undeniable pattern. And it also comes up with reasonably correct dates for historical events that we actually have eyewitnesses for. So you can't completely throw out carbon-14 dating. So what he does is he takes carbon-14 dating and basically puts it on a scale where over the last 2,000 years, it's relatively been constant in terms of the decay rate of carbon-14. And then in the past, the decay rate was higher and higher and higher and higher. So it's sort of an asymptote. All right. And so what happens is that a date that's 4,000 years old for carbon-14 dating is probably more like 3,500 years ago. But a date that's 7,000 is like 4,000. And a date that's 10,000 is like 4,200 years old, and so forth. By the time you get to 50,000 years old, that represents in his model basically something that's very close to the arc. All right, so that's how he smooshes it all into just 4,300 years. So now what I would say is, okay, great, let's do that. Let's just squish it all in. If you do that, you still have the elephant that's 700,000 years old, the mammoth that's 700,000 years old, being right at the flood. And then you have mammoths that exist over time all the way up to just under 4,000 years ago, which he can't say is 4,000 years ago. He's going to have to say it's more recent. And he's going to say that that's more like 2,000 years ago. So this is over 2,000 years, but still represents a lot of generations of elephants. And over those generations, there hasn't been a loss of genetic diversity, right? The total diversity of those elephants hasn't changed very much. All right, and the last thing I'll say that is really a complete failure on the part of Jeanson to appreciate is that we're talking about dozens and dozens and dozens of genes that are affected. All right, the list is longer than is shown here, but if you remember, all the names, the names in these boxes represent genes. And inside those genes are multiple different sites that are variants that are being selected for. All right, and multiple of these genes are working together to form physio traits. So now, no young Earth creationist really ever models this in terms of how many generations this would take. If you had all these variants mixed together in a common ancestor, so here's a bunch of, if his model is right, that God created all these particular variants, these things that are fixed in the woolly mammoth were just one variant among many variants that existed in a population of the original elephant that was on the ark. Right? If you ask yourself, all right, now a population that has all this variation starts to move north, how long would it take? How many generations would it take for you to select for these specific variants in all these different genes for all these different traits? That's the whole idea of this polygenetic selection. All right, polygenetic selection is you're selecting for a trait that's better in the cold, but you're having to select for many genes at one time. But in one elephant, you might have, well, I got the good gene in this elephant, but for this other gene, I have not a very good variant. And another elephant has the other variant that you need, and there's a different elephant that has the other variant you need. Right? It's not just one generation. It's not one elephant surviving and having offspring that suddenly you'll have all these different variants that are all packaged together in one elephant. This requires a tremendous number of reproductive events with selection occurring consistently across many different genes. And you have to have recombination of these genes in order to link them together. So you have to have a lot of reproduction occurring in order to link the genes together that are the positive alleles and then sweep out all the other ones in order to become fixed. I challenge young Earth creationists to take a polygenetic model in which you're working with several dozen alleles, several dozen variants on different chromosomes in different positions, and sort them and have them in a varied population, and then do the modeling to show how many generations it would take to combine all those into and fix them into a population like this. I tell you what the answer is going to be. The answer is going to be it's going to take tens of thousands of generations, and elephants have a 20-year generation cycle. All right, so we're talking 20 to 100,000 years in order to just take a variant pool of a genetic pool of variants in an original population and to sort them out through the process of natural selection. Just to sort them out just for natural selection to sort them out into a consistent package of individuals that are in a separate population is an extremely long process. There's no quick way of doing that. So if they're going to appeal to, as Jeanson does, to forces like natural selection as the thing that selects for and sorts heterozygous traits and turns them into homozygous traits, it's a very unrealistic model. Mathematically, not even close, right? It sounds all nice in the foo-foo world of waving your hands and saying like, oh yeah, I can show like that generally this happens. But when you get talking like the nitty-gritty of it, of actual numbers, number crunching and modeling that, it falls apart. Right. So failed predictions on a variety of different levels is the model of created heterozygosity and natural selection. Right. That's the name of his model. Right. Created heterozygosity. God created all the heterozygosity, the variation, and then used natural selection to sort that heterozygosity. Doesn't work on the timescales that are required for Jeanson. And that's not even to mention the problem of a bottleneck for the flood, starting with only 2 individuals, etc., etc., etc. All right, I think that's, I have worked this particular paper to death. Yeah, this was a fun paper. I enjoyed this. This is really neat. I mean, it's just fascinating to find that we can get data from things that are extinct today. And this type of data is just rolling in now. We're getting better and better technology for pushing back the boundaries in terms of the ages we can find things at. We're finding new ways to get samples out of tissues that we weren't able to get data out from before. So this is going to be like a daily occurrence where we find out really cool stuff about ancient organisms. And so it's a chance to test these models. Jeanson also has this whole trace thing with human Y chromosomes, and we're sequencing more and more ancient Y chromosomes. And so his models are going to be tested. I would say they've already been tested, and he's already failed there as well. But he's going to get tested more in the future. And it'll be very interesting to see what kind of response comes from that. All right, let's call it quits there. Thanks for hanging out with me. That's more Pachyderm party time. I'm sure I can come up with, I got a whole bunch more things that I've written about elephants and young earth creationism. So more to come. Till then, we'll see you later. Bye-bye.